Our mission, as always, is to take a stack of sources, sometimes a really dense stack, and pull out the core knowledge you need. And today, we're taking on something huge. Basically, the biography of cancer itself.
Yeah.
We're going to try and trace its story, right, from, ancient history, whispers of it, all the way to the molecular enemy we face now.
It's really the only way to approach it, I think. Because how we define cancer, how we treat it, it's always mirrored our deepest understanding of the body.
Or misunderstanding something.
Yeah, or misunderstanding. And you see how the search for a cure, it's actually driven some massive breakthroughs in biology, medicine, even politics, really.
And the context here, it's just immense. We are talking about the defining illness of our generation. The numbers alone are pretty sobering.
They really are.
In the U.S., it's something like 1 in 3 women, 1 in 2 men will develop cancer. Globally.
Oh.
Well, it's described as this lethal, shape-shifting thing, always adapting.
And thinking of it as a biography, trying to understand its, let's say, personality, it helped us ask that big question. Can we actually genuinely conceive at ending this?
Or is it just becoming, the new normal? A heavy thought to start with.
It is.
Okay, so the biography. Chapter 1 has to be the ancient world, right?
Right.
When it was barely recognized.
Right at the beginning, yeah. The absolute earliest record we sort of have is from Egypt, a physician named Imhotep, around 2500 BC.
Wow, that far back.
Yeah. And he described what sounds like a breast tumor, a bulging mass, but the crucial part, his note on treatment.
What was it?
Basically, there is none. Just nothing to be done.
That sense of helplessness.
It lasted for centuries. I mean, Hippocrates later, he gave it the name Carcinos the crab because of how the veins seemed to spread out.
Still use that root word today, carcinoma.
We do. But it was Galen, maybe 5 centuries after Hippocrates, who really locked in the dominant theory for, over 1,000 years.
Ah, this is the humoral theory.
That's the one. The body governed by 4 fluids, or humors: blood, phlegm, yellow bile, and black bile.
And cancer was?
Cancer, according to Galen, was caused by an excess of black bile, melancholy, basically.
Okay, so a fluid imbalance. But what did that mean for treatment? It sounds like you wouldn't just cut out a lump if it's a fluid problem.
Exactly right. And this is where it gets quite profound and, frankly, destructive. Galen's advice was that cancer was actually best left untreated.
Wait, really? Leave it alone? Why?
Because the black bile, the cause, was seen as systemic. It was everywhere in the body.
Well,
Cutting out the tumor, the local manifestation, was, in his view, pointless. Worse, actually. He argued surgery could make it worse. You disturb the area, the black bile just pools there, maybe turns a quiet lump into an ulcerating wound.
So the theory led directly to inaction, or even a fear of intervention.
For more than a millennium, yeah. Fatalism, isolation. Think about Queen Atossa, the Persian queen, around 500 BC.
That's her story.
She developed a breast tumor, hid it, shame, isolation. The usual response, maybe amplified by her status. Eventually, she got desperate.
Yeah.
She had a slave and not a physician cut the tumor off with a knife, just hacked it off.
Good grief. That speaks volumes about the desperation and the lack of options if the official medical advice was do nothing.
Absolutely. It shows that profound isolation, when the system tells you the problem, is this pervasive, untouchable fluid imbalance. But, theories eventually bump up against reality, and the humoral theory started to crumble with the rise of anatomy.
Someone actually started looking inside.
Yes, Andreas Vesalius, 16th century. He did these meticulous human dissections, a pretty radical thing back then.
And I bet he was looking for that black bile Galen talked about.
He was looking for all the structures Galen described, and the crucial thing, he couldn't find physical evidence of black bile as like a separate fluid causing tumors. He found organs, tissues, blood, but no distinct black bile reservoir.
So observation starts chipping away at the old theory.
It laid the foundation for a whole new way of thinking. Pathology, focusing on physical changes in organs and tissues, not these abstract fluids. Cancer started to look like a local problem, a structural thing.
Okay, so the thinking shifts, but to actually act on that, to start, the age of the knife he mentioned, that needed more, right? Surgery was brutal.
Oh, unbelievably brutal. Before the mid-19th century, patients often died from the shock and pain of surgery itself, or almost certainly from infection afterwards.
Right.
The game changers were 2 huge breakthroughs. Anesthesia ether, mostly, at first took care of the pain, and antisepsis lister, carbolic acid, clean practices dealt with the infection.
Suddenly, surgeons could actually operate.
They were unleashed. Between roughly 1850 and 1950, surgeons could, brazenly attack cancer, as one source puts it, cut open the body, go deep, remove the growth. It was the era of the local war.
And the peak of that local war strategy, that has to be William Halsted and his radical mastectomy, doesn't it?
Absolutely. Halsted was, well, almost zealous. He believed, truly believed, the knife could cure cancer. You just had to be radical enough. Cut out everything.
Describe how radical it was.
It was technically brilliant in a way, but incredibly disfiguring. He didn't just remove the breast tissue. He took the underlying chest muscles, the pectorals, and all the nearby lymph nodes in the armpit. Everything.
The idea was total eradication at the source, uprooted completely.
Exactly. He was utterly convinced he was exterminating cancer at its root. That was his phrase.
But the conviction didn't match the reality, did it? What did the long-term data show?
That's the tragic irony. It was a profound failure, really, for the purely local view. Despite the surgical skill, despite the radical approach, the statistics were just grim. Nearly half the women treated with Halsted's radical method still died of breast cancer within 3 years.
So even if you removed every visible trace with incredible precision, the cancer still came back somewhere else.
Precisely. It proved definitively that the disease was often systemic long before it was detected. Just like Galen had argued, ironically, but for entirely different reasons.
Not black bile, but cells that had already spread.
Micrometastases,
Yeah.
cells that had escaped the original site. Halsted's failure was actually a crucial piece of evidence. It showed surgery alone wasn't enough for many cancers. The war had to become systemic.
Right. So that realization that cancer often wasn't just local, it forces a shift. We need treatments that can travel through the whole body. Enter the chemical paradigm.
The search for the magic bullet.
The magic bullet, yeah.
Yeah.
Paul Ehrlich was influential early on. He worked with dyes, aniline dyes, and showed they had specific affinity. They'd stick to certain cells or bacteria, but not others.
So the idea was, could we find a chemical that sticks only to cancer cells and kills them?
That was the dream. His own search for a cancer cure didn't pan out, but the concept was powerful. And the first real clues about chemicals that could attack cancer cells came from, well, a horrific source.
Chemical weapons, yeah. Nitrogen mustard from World War I. Awful stuff. But researchers studying its effects noticed something. It absolutely decimated bone marrow cells.
What?
And lymph nodes.
Fast-growing cells.
Exactly. Rapidly dividing cells. And that sparked an idea, a dangerous one, but pivotal. If a poison targets rapidly growing normal cells, maybe it could target rapidly growing cancer cells even more effectively.
Using poison as medicine. But how do you even track if that's working inside the body, especially with a solid tumor you can't easily see?
That's a huge problem, measuring success. And this is where Sidney Farber comes in, with a really critical strategic insight in the 1940s. It wasn't just about finding a drug, it was about finding the right disease to test it on.
Okay, what did he choose?
He focused on childhood leukemia, specifically acute lymphoblastic leukemia, ALL.
Why leukemia?
Because the cancer cells, the leukemic blasts, they weren't hidden in a solid lump. They were floating free in the bloodstream.
Ah, so you could actually draw blood.
And count them. Under a microscope, you could literally count the cancer cells before treatment and then count them again after giving a drug.
He made the disease countable, quantifiable.
Precisely. It transformed the approach. It wasn't just guesswork anymore. It became a measurable experiment. You could see, objectively, if your intervention was killing cancer cells.
Did it work quickly?
Relatively, yes.
Yeah.
In the late 1940s, Farber started using antifolates. Chemicals like aminopterin, these were derived from that same toxic principle interfering with cell division.
And the results in these children?
For the first time ever, he achieved remissions. They were described as flickering and feeble at first, often temporary, but it was proof that a chemical could actually push back this previously hopeless disease, the first real crack in the armor.
That must have been electrifying. But Farber knew science wasn't enough, right? He needed public support.
He was very shrewd about that. He teamed up with Mary Lasker, a formidable advertising executive and health advocate. She was determined to make cancer a national priority, like polio had become.
How did they do it?
They used the story of childhood leukemia, the pathos, the potential for cure, very effectively. They launched huge fundraising campaigns. The famous Jimmy Fund started then, using radio celebrity.
Making it a public cause, not just a private tragedy.
Exactly, turning a medical crisis into a goal-driven public movement. And Lasker especially pushed this into the political arena.
Leading to the big one, the National Cancer Act of 1971.
That was the culmination. Nixon signed it. It officially declared a federal war on cancer. framed very deliberately, like the Apollo moonshot program, a massive injection of government funding and resources, all aimed at one goal: conquering cancer.
So the war on cancer begins, resources pour in. But the real revolution, arguably, came when the focus shifted from just poisoning fast-dividing cells to understanding why they were dividing uncontrollably in the first place, the genetic level.
Absolutely. We moved from sort of carpet bombing to trying to understand the enemy's command structure. The modern definition is basically this: cancer is caused by mutations, changes in the DNA. These mutations break the intricate genetic circuits that normally control when a cell divides, when it grows, and crucially, when it dies. Lose that control, and you get runaway growth.
And scientists started identifying the specific genes involved. I know the analogy often used is a car's accelerator and brakes.
It's a really helpful analogy. Let's start with the accelerator. There are normal genes in our cells called proto-oncogenes. Think of genes like RAS or SRC. Their job is to tell the cell, okay, time to grow and divide now, when appropriate.
Like pressing the gas pedal gently when needed.
Exactly. But a mutation can happen that turns that proto-oncogene into an oncogene. That's like the accelerator pedal getting jammed to the floor. The cell gets a permanent, unrelenting signal to go, divide, now, even when it shouldn't.
Okay, so that's one way cancer starts a stuck accelerator. What about the brakes?
The brakes are the tumor suppressor genes. The big breakthrough here came from Alfred Knudsen and his two-hit hypothesis.
2 hits, meaning
We inherit 2 copies of most genes, one from each parent. Knudsen proposed, and was proven right, that for many cancers, you need to disable both copies of a specific tumor suppressor gene. You need 2 damaging hits to completely lose the braking function.
like needing both the main brake and the emergency brake to fail before the car runs away.
That's a great way to put it. And the most famous of these brake genes, probably ARB, involved in an eye cancer, and the absolute titan, P53. It's sometimes called the guardian of the genome.
Why is P53 so important?
It senses DNA damage. If the damage is repairable, P53 halts cell division until it's fixed. If the damage is too severe, P53 triggers apoptosis programmed cell death. Basically, it tells the cell to commit suicide for the good of the organism.
So if you lose both copies of P53,
The cell loses a critical safety mechanism. It can accumulate mutations, ignore damage signals, and keep dividing. It's a massive step towards cancer. And this 2-hit idea also helped explain why cancer risk increases so much with age.
because it takes time to accumulate those 2 separate hits, those mutations, in the same cell line.
Exactly. It's a game of probability and time.
Okay, so we've mapped the circuitry, the oncogenes, accelerators, and the tumor suppressors, brakes. How did that knowledge change treatment? This must be where targeted therapy comes in.
This is the paradigm shift. Moving away from chemotherapy, the fairly indiscriminate poisons that just kill any rapidly dividing cell, including healthy ones, towards precision strikes.
Aiming for the specific broken part.
Right. And the poster child for this is probably the story of HER2 and breast cancer. Researchers found that in a specific subset of breast cancers, about 20–25%, the cells had way too many copies of a particular oncogene called HER2.
Amplification. The accelerator wasn't just stuck, there were dozens of accelerators.
Exactly. These cells were utterly dependent on, almost addicted to, the constant grow signal from HER2. One researcher described them as hypnotically drunk on the oncogene.
That paints a picture, so
Yeah.
the target was obvious.
It was. And scientists developed a drug, an antibody, called Herceptin trastuzumab is the generic name. It's designed specifically to find and block that overactive HER2 protein on the surface of the cancer cells.
A guided missile, not a bomb.
Pretty much. And for women with that specific HER2-positive type of breast cancer, like Barbara Bradfield, who was one of the early trial patients, the results were just stunning.
What happened?
They saw palpable deceleration of the tumors. Sometimes they literally seemed to melt away. It wasn't always a cure, but it significantly extended lives. It was profound proof of concept. Identify the driver mutation, target it precisely, and you can make a huge difference.
It really is an incredible arc when you lay it out like that, from thinking cancer is what, bad black bile,
Yeah.
to designing an antibody that latches onto one specific malfunctioning protein on one type of cancer cell. The journey is just mind-boggling.
The progress is undeniable. It's immense. You know, that thought experiment, if Queen Atossa lived today, with modern surgery, radiation, maybe hormonal therapy, maybe targeted drugs like Herceptin if her tumor was right, she could potentially gain decades of life. 17 to 30 years, some estimate.
That's extraordinary. But as you said at the start, the war isn't won. What are the big hurdles now? even with targeted therapies.
Well, I think there are two huge challenges, broadly speaking. First is the ongoing biological challenge.
Meaning,
Meaning we found her too, yes, but cancer isn't one thing. It's hundreds of diseases at the molecular level. The Cancer Genome Atlas project is trying to map all the mutations across all cancer types.
creating a complete map of the enemy's territory.
Kind of. It's sometimes called mapping the 13 mountains of cancer's complexity. It just shows the sheer scale. We might need hundreds, maybe thousands of different targeted therapies to cover all the bases, and then there's resistance, evolution within the tumor. It's biologically incredibly complex.
Okay, that's the biological mountain range. What's the second challenge?
The second is, arguably, even tougher. It's the social challenge, specifically prevention.
The one that gets less fanfare, maybe less funding.
Often, yes, but the potential impact is enormous. We have historical proof. Think back to Percival Pott in the 18th century.
The chimney sweeps.
Exactly. He noticed they got scrotal cancer at vastly higher rates. He linked it to soot exposure. Clean up the work conditions, the cancer rates drop. Simple, powerful prevention.
Or the huge one, smoking and lung cancer.
Dahl and Hill, mid-20th century. Their work definitively linked smoking to lung cancer. Public health campaigns, taxes, restrictions, smoking rates went down, and eventually lung cancer rates followed. Massive gains from prevention.
So why is prevention still such a struggle today when we know so much more about risk factors, diet, obesity, environment, infections?
Because, as the sources put it, prevention forces us to confront the customs, rituals, and behaviors that are at the very definitional cores of our lives.
It's easier to develop a pill than change how society eats or works or pollutes.
Sadly, often yes. Changing deeply ingrained societal patterns, tackling environmental exposures, addressing health disparities, that's incredibly difficult politically and culturally. But arguably, that's where the largest potential gains against cancer still lie, not just in the lab, but out here in how we live collectively.
That's a really powerful point. And it ties back to that sobering statistic we started with. If cancer rates are creeping towards 1 in 2 in some places.
Then the illness really might be becoming the new normal.
Yeah.
And maybe the perspective has to shift again. Perhaps the primary question isn't just if we, as individuals or as society, will encounter cancer, but when.
And how well prepared we are, both biologically and socially.
Exactly. How well can we detect it early, manage it smartly with the tools we have, and crucially, how much will we invest in preventing it in the first place? It brings us back, in a way, to needing a systemic solution, just like Galen thought. Though our reasons now are based on genetics and environment, not black bile.
A truly sweeping history and some really vital perspective for today. Thank you for taking us through that deep dive.
My pleasure. It's a story worth knowing.
Indeed. We'll be back next time with more crucial knowledge. You need to be well informed.